Finally, part 3 was a test cross. So test cross is when you want to determine what is the genotype of an organism that is showing the dominant features. Is it heterozygous or is it homozygous? So there's two possibilities. Either this unknown is homozygous dominant for both of the traits, or it's heterozygous for both of the traits. And so that was what you had to first project. So you were given a corn, experiment 3, and you had to figure out what its genotype was, and you first had to have a guess. So to figure out a genotype which is showing the dominant, you need to cross it with a known genotype. And in this case, we always use homozygous recessive for both the traits. So you can see in the background information, it gave you an example of what would you expect if it was homozygous dominant and you crossed it to homozygous recessive for both traits. They would, all of the offspring would be homozygous dominant. If it was a heterozygote, then you would expect a ratio of 1 to 1 to 1 to 1 of each of the different possible phenotypes. And so that's what you had to set up in your test cross. So if we look at a sample that somebody did in the class, if you count how many kernels they looked at 155. They observed 38, 33, 44, and 40 of the different possible phenotypes, which are your combinations of purple smooth, purple wrinkled, yellow smooth. What did you actually expect? Well, the expected is if I'm expecting a 1 to 1 to 1 ratio. So therefore, 25% of each of them would be those phenotypes. So 25% of 155 meant that I should have got 39 using this number that the student calculated. Each of them should have been 39. So there's your expected values, and now you can go and plug in for your chi-square test. So if you look at what the student counted, she was kind of close to each of them being 39. So now you would plug in each of those ratios in your chi-square test. So observed would be 38, subtract 39, square that, and then divide by 39, and that would be for your first one. And then you would do another value for the second one. I got 33, subtract 39, square that result, then divide it by 39. And so on. And that way you would calculate what is your chi-squared value. And if you got a significant result, if you could accept a result at greater than 95%, then your hypothesis would be right. If you got this 1 to 1 to 1 ratio, then you are expecting this individual corn that you're looking at, the genotype of the parent was mostly heterozygous and not homozygous. So test crosses are really important to figure out genotypes of the parent when it's unknown. And in order to do that, you cross it with a known. And our recessive traits, we always know what they are. Remember, to be recessive, you have to have 2 little letters at both of the loci that you were looking at. So AAPP would be a homozygous recessive, or in our case, a little p for being yellow, and little s for being wrinkled instead of smooth is the homozygous recessive trait. So hopefully this helps you figure out what you did incorrectly in that lab. And as I said, I gave you all credit, but go back and make sure you understand your ratios. 9:3:3:1 for a dihybrid cross, and 3:1 for a monohybrid cross, and be able to answer those are important when you're doing these genetics problems. Good luck. Goodbye.